Hello, everyone. Thank you for joining our webinar today. So I'm going to talk a little bit about proximity ligation technology in genome assembly. First, a little bit about us. And this does not want to work. Now, here we go. Oh, yeah, maybe I should just mention it beforehand. My internet is wonky throughout the day. So if I'm suddenly gone, then just give me a minute to log back in. It's just a general problem where I am right now. Anyway, so first I just wanted to tell you a little bit about Dovetail Genomics if you haven't heard of us. So originally Dovetail is a company offering genome assembly as a service. About last year, we launched a kit as well that can be used for genome assembly. And since then, we launched additional kits all around proximity ligation that can be used not only for genome scaffolding, but also for epigenetics and chromatin conformation. We are located in Scotts Valley in California. If you don't know Scotts Valley, that's fine. It's south of San Jose, which is close to San Francisco. If you're in the area and you're interested, you're welcome to come by. So this is what Dovetail offers for products and services. So for products, as I mentioned, we launched a kit not too long ago. We're calling it Omni-C. It's basically a high-C kit that uses DNAs instead of restriction enzymes. And this one we mainly use for genome assembly. And this is what this talk is going to focus on. In addition to that, we have other kits like a micro-C kit, which is basically high-C using MNAs. It is better for chromatin conformation and loop calling. And also we have a high chip kit based on that technology. Apart from a kit, we offer services as well. So, and most people in the genome assembly world know us from there. So we do de novo assembly, genome scaffolding, and I just have a picture. We're currently running a grant promo. So we extended this just for 2 weeks. If you would like to apply, we can do this. Also, we have currently a promo for the kit if you're interested in trying it out. So the talk is split in 2 parts. So the first part is going to be like very basic. How does proximity ligation work? How does this play into genome assembly? And the second part is what can you do with this kind of data set? So I can already tell you the whole thing will take about 40 to 45 minutes. If you have questions, you can either post it on Slack or on the other chat website, or send me an email. I had my email address there. I'll show it at the end again. But first of all, what is proximity ligation? So if you're looking at the DNA, so this is an example for chromatin. Proximity ligation means we're looking at connections on DNA. So usually if you look at a sequence, it looks linear, but obviously in real life, DNA is not linear, it's jumbled up in a ball, and things are very close to each other. And this is what we use for genome scaffolding. When the company was founded, we had this method called Chicago, which is basically an in vitro version of high-C. In terms of scaffolding, it works on a similar level as 10x. It is a proximity ligated library like high-C, but as input material, you use high molecular weight DNA. We introduced HiRISE, which is our scaffolding software, similar to SALSA or 3D DNA, but HiRISE is proprietary to Dovetail. But last year, we actually introduced Omni-C, which replaced Chicago completely and high-C. So now instead of creating 2 libraries for genome scaffolding, we're only doing 1. And because of the way Omni-C works, we are able to phase, for example, genomes much better, or do SNP calling. And I have some data for that as well. But first of all, just as a recap, some of you already might know this. How does high-C work, and how does Omni-C work? What are the differences in between these 2? So high-C first. So what you usually do to do high-C is you take tissue as input material. This could be leaves, blood, or anything. You cross-link the DNA with the proteins. So just look, this is a piece of DNA. See 1 and 2 at distal ends of the DNA. Oops, sorry. So you cross-link everything. So you're basically locking the DNA in 3D space. Then you take a restriction enzyme, you digest the DNA, then you end repair it and biotin label it. And then this is the important step. You ligate everything together that's in close proximity to each other, meaning 1 and 2 that used to be at distal ends are now ligated together. After that, you reverse the cross-linking, you remove all the proteins, you clean everything up, and you're basically getting an Illumina library where read 1 and read 2 map back at 2 different locations of the genome. And it basically looks like this. So if you have your reference genome down here, and you create your Hi-C libraries, and then you take your Hi-C libraries and plot it against your reference genome, you will have a certain distance. So this one here, for example, with 1 and 2 would map back here and here. And of course, statistically, you will have way more reads that have that have less range information, so are closer together. So that means the lower you go here, the more reads you will have. So every time you see this Hi-C reads mapping back against the reference, you always have to think in these triangles. So it always goes down like this. And this is basically how it looks like. So this would be 2 chromosomes, chromosome A and chromosome B. And all these dots are basically Illumina read pairs. Every dot maps back at 2 different locations. So this dot would map back here and here. This one would map back here and here. And the higher up the dot is, or the read pair is, the larger is the read pair separation. As you would expect, if you cross-link a chromosome, you're basically binning the data. So that means most of the reads you will be within the chromosome. Some of the reads will be between chromosomes. These are actually quite interesting. So these show you, for example, in a nucleus, if which chromosomes are next to each other. You can actually determine the location of the chromosome in relation to each other. There was a very nice talk last year I saw that showed, for example, that the closer the chromosome is to a nuclear membrane, or the more it is expressed, the closer the chromosome is to the nuclear membrane, which makes sense because then the transcript doesn't have to travel that far. But we are using this kind of information not necessarily for chromatin conformation, which you can do. We are using it for genome assembly. So this would be a draft assembly, and you can see every of these boxes you see here is a scaffold. So you can see this one is quite fragmented. So we're having a draft assembly, ideally something with long reads. We have our Hi-C data set, and then we're going into our pipeline. So in this case, this pipeline is HiRISE, which is our proprietary pipeline, but all pipelines more or less work similar to that. So what happens here is we're using the Hi-C data set to basically put together all these scaffolds, connect them, and then get much, much larger scaffolds, so much so that one scaffold ideally is actually 1 chromosome. So how does this actually look like when you do this? So let's say this down here is the draft genome. All these colorful blocks here is the reference assembly. And this is the Hi-C data you're plotting against this reference you created before. So as I think in triangles again. So every of those read pairs maps back at 2 different locations, and this is exactly what you would expect. So the density of the reads are higher the closer they are. So up here, that would be, let's say, 150 megabases apart. These would be 1 megabase apart. So obviously, you have way more here. But sometimes you get something like this. This is basically read pairs that float in the air. So like this bow tie kind of structure. And this, you would not expect this. So the software would see that and say, okay, something is wrong here. These belong to this contact here. So the HiRISE would take this, change the orientation of that contact. All these read pairs are falling back into these gaps, and therefore the orientation of that contact is corrected. So this is how it generally works to correct the orientation of a draft assembly. So how is Omni-C now different to that? Because basically what I just showed you, Omni-C dataset is a Hi-C dataset and does exactly the same thing, but there are some differences. So first of all, the first step is still the same. We're cross-linking everything, but then instead of using restriction enzymes, we are using DNAs to digest it. So this has well, this has an impact on first of all, on the kind of fragments you're getting here. First of all, you don't have sticky ends anymore, so you don't do end repair, and you don't label these ends directly with biotin. Instead, we are ligating an oligoadapter in there that is already biotin labeled. But otherwise, it's still the same. So we are ligating this in there. We are removing the cross-linking, reversing the cross-linking, and we're getting an Illumina sequencing library. The advantage of doing that is what happened usually during these ligation steps. So here is restriction enzyme based Hi-C, and here is DNA based Hi-C. Usually you can get 3 different outcomes from doing this ligation step. So first of all, you can get a ring, so it self-ligates. You get religation with the original partner, or you're getting, and this is the one you're actually looking at, is you're getting this range information. So for example, these 2 that are further apart, now they are together. The nice thing is using DNAs, first of all, because now we're not biased towards the restriction enzyme motif, the digestion usually gives you a smaller fragment size. The second advantage of the smaller fragment size is you do not need to use mechanical shearing with the Cuvaris in this protocol because the fragment size is already okay. The second thing is we do not get self-ligation events. So that means the data set that you're getting out in the very end is cleaner. So that means the sequencing depth that is basically eaten away by these ones you don't want, we don't get them in the first place. Of course, you're still getting the religation events, which might actually give you some information as well. And of course, the valid distance ligation, so to say. The workflow for this is just real quick. The workflow for Omni-C. This is the kit. It's a 2-day workflow. Day 1 is mainly the DNA, the Hi-C part. Day 2 is just the DNA library prep. We have a lot of QC steps in here and quite a few stop points if you don't want to do this in 2 days. Honestly, the first time you're doing this, you might want to stretch it out for a little bit longer as well. Also, if you would get a kit, you are actually getting a scientist included. So you would talk to someone from Dovetail who will guide you through the protocol. You can send them the QC data. They can say, okay, this looks good or this doesn't look as good, and then you might have to repeat something or not. But there is also enough reagent in these kits to try several things. Yeah, but this is now how the data set actually looks like. So in this case, we are comparing restriction enzyme free Hi-C, so Omni-C, with Hi-C using a single restriction enzyme, which is mostly the homebrew versions, Hi-C with multiple restriction enzymes, which are some commercial kits. And in this case, we are just plotting the data back against the reference. And what you can see here is there are certain gaps. This is mainly because if you're using a restriction enzyme to create your Hi-C libraries, you have a bias toward that restriction enzyme site, meaning you're going to get a pileup of reads when you sequence very close to the cut site. The further away you go from the cut site, the less coverage you're going to have. So meaning, for example, in these areas here, these are just far away from restriction enzyme sites. So that's why there's less coverage. With multiple restriction enzymes, it's better, of course, because you have just more cut sites in the genome, but you will still have certain areas, especially repetitive areas, that are very poor in restriction enzyme sites. While if you're using Omni-C, so DNAs, since it does not have a restriction enzyme motif, you get a way more uniform digestion and therefore a more uniform coverage across the genome. So how does this actually look like? So if you're plotting your Hi-C or Omni-C reads back against the reference, so this is human data. It's similar to what I showed you before. So the reference is down here. All this orange stuff is basically Hi-C. So this is a content matrix. When you quite often see these white lines here, and these white lines are basically areas without any data, meaning there is no restriction enzyme sites here. So there is not enough data to plot it back against the reference. And this is just a few examples on different chromosomes. If you're looking at Omni-C data, it looks way more crisp because we just don't have this bias anymore. But how does this now? So this makes sense, of course, if you want to do genome confirmation. So you can already see you're getting additional data you haven't seen before. But why is this important in genome assembly? And this has to do with the way genome assembly or genome scaffolding works. So typically or historically, you needed, and if you want to scaffold a draft genome with Hi-C, you should have a input N50 from about a megabase. So you'd need a quite contiguous assembly to begin with. The reason for that is you can only scaffold a contig if there is Hi-C information, meaning you can only scaffold it if there is a restriction enzyme site on that contig. And of course, if you have an N50 of a megabase, statistically, because the contigs are so large, you're going to have a restriction enzyme site on it. And if you're comparing this, so blue is Omni-C, gray is Hi-C, and here is the number of restriction enzyme sites or cut sites basically per contig. So if you don't have a cut or no restriction enzyme site on your contig, you don't have much information to scaffold. So you need one, ideally 2 or 3 cut sites, so you have information to actually scaffold it. While with Omni-C, because you're using DNAs, this is not necessary because you will statistically always have a cut site somewhere on the contig. So there will always be information to do that. So if you're coming to us, for example, as a service, because of that, we are asking now for not an input at 50 of a megabase to go into a Hi-C pipeline, but for us, it's enough already to go to an 50 of 100 kilobase, and we are able to scaffold it with Omni-C. And I have some data for that I'm going to show you in a second. But just as an example, what happens if you try to scaffold a genome with a more fragmented input assembly with only restriction enzyme-based Hi-C? So this is weeping lovegrass. It's done with a PacBio assembly. So it has an input N50 of 380 kilobases, which is not too bad, to be honest. So if you go into Hi-C, or if you use Hi-C alone, you scaffold, you get an N50 over the 5 megabase, which is not too bad, but obviously not chromosome level. And because of that, as I mentioned before, Dovetail had this intermediate scaffolding step called Chicago. So it improved the draft assembly from about 380 kilobases to about 800 kilobases. And then if you use the same Hi-C data set on top of that, it was able to go up to chromosome level. With Omni-C, we don't need to do this anymore. So we got, even as a service, we don't do Chicago, we directly only go into Omni-C. And this is some data for that. So this is just to show you that the performance of Omni-C alone is just as good as Chicago and Hi-C with a good input N50. So these genomes, as you can see here, the draft assembly is already very, very good. So this plant here, the cheatgrass, has an N50 of 27 megabases, mouse 10 megabases. We have here an aphid 2 megabase input, and Chicago and Hi-C or Omni-C more or less scaffolded to the same level. And it works quite well. But if you're looking at a more fragmented genome, so these are now all genomes with an N50 lower than 1 megabase. You can see a 670 kilobases, 470 kilobases, 140 kilobases, 200 kilobases. Just so you know, these are all PacBio input assemblies. The mollusk down here is a hybrid assembly, so PacBio and Illumina data. Chicago and Hi-C together do a good job, but only Omni-C alone can scaffold just as well. So you don't need any intermediate scaffolding steps. So you can see even in this case for the reptile, Omni-C alone is actually better than Chicago and Hi-C. And one of the reasons is for this one, there was a large stretch where there was no restriction enzyme site, which could not be assembled because of it, but Omni-C was able to do so. So this is basically a bragging site. So I mentioned before, if you would come and do this as a service, we are asked, and you already have a draft assembly, we would ask for a draft assembly with at least 100 kilobase and 50. However, we had a few projects where we couldn't get there, and this was mainly due to lack of material. So there was just not enough a material to do a long read assembly. So these are all Illumina draft assemblies. So there is a mammal with 80 kilobase N50, a plant with 40 kilobase N50, and this was pretty bad because it's protected. It's a 5 kilobase N50 input assembly. And Omni-C alone, without any intermediate scaffolding step, went from 5 kilobase to 140 megabase, which I know sounds unbelievable, and I will give you that, because I'm very sure if you're looking into this assembly in a bit more detail, you will find small contigs that are in the wrong orientation and are just not scaffolded properly. That's why this is red, and I might next time do it, maybe it should blink, I think, because if you would come to us as a service and you would say you have this one as an input assembly, we would tell you not to do it because we would not trust that this works well. It is mainly if you have something and there's just no other way, you can get something out of this, but it's absolutely not recommended. Oh, the one thing I might have to mention here as well. So all the data you saw here is done with our HiRise software, which is proprietary to Dovetail. If you're using any of the other open sources like 3D-DNA or Salsa, of course, the assembly might differ. I talked to Jay Guria, who is the author of Salsa 2, so he already told me he added a DNA switch to the software, so you're not dependent on the restriction-enzyme sites or anything like that when you're using his software for assembly. Okay, now I'm just going a little bit into what else Omni-C can do. As I mentioned before, Omni-C is basically just another type of Hi-C library. So everything you can do with a Hi-C library, you can do with Omni-C as well. But because it has this very uniform coverage, it can do a few things better, or you can do a few things in the first place that you can't do with a normal Hi-C library. So, and I haven't actually mentioned it before, but you can see this dotted line here, and the dotted line is actually the distribution of a shotgun library. And if you're looking at the distribution of the Omni-C library, you can see it is very shotgun-like. While with restriction enzyme-based Hi-C, basically what you have here is the bias towards the restriction enzyme site, so it has nothing much to do with a shotgun library. So you could actually look at an Omni-C library as a single read whole genome library. Single read because between read 1 and read 2, you have a certain distance. They're mapped back at 2 different locations on the genome and you just don't know where. But if you're just looking at it as single read, it's basically a whole genome library. So we tested a few things to do with this. So first of all, we went into Hi-C Break Finder, which is a software you can use to detect structural variations. This is something you can do with all Hi-C libraries, of course. We actually ran it through GATK for SNP and ILNL calling. This you cannot do with normal, or you should not do it with a normal Hi-C library. But because we have it so shotgun-like, it works actually quite well here. Of course, genome assembly, and I talked about this before. And because we get all the SNP information, we are very good in phasing, and I have a slide for that as well. And of course, you've seen it before, because the data is so much more dense, it is significantly better to use this data set for TAT calling or just chromatin confirmation in the general. So I'm just going through a few of these things that you can do with it. So here is structural variation detection. Basically, this is exactly the same slide as I showed you before. But now just imagine that instead of having different contexts here, so let's go with the human genome because it's easier. So let's say you have the human reference genome, and that's your reference genome. Now you take a data set from another human and plot it against your the reference here, and you get this. Then this is not a misassembly because your reference is okay. This would be a inversion. So that means this is actual biological effect. It's a structural variation. And this is what people use Omni-C data as well for. So we have a few kind of population studies. So we created a draft assembly from one individual. I think it was it was an aunt project. So we created a draft assembly from an aunt, and then someone created several Omni-C libraries just from additional aunts and mapped them back against the reference we created, and then just looked for these structure variations here, just to see the differences in the genome. So this is a data set you can get with the Omni-C data as well. It helps, for example, if you buy our kit, it comes for 8 samples. So you can use one of these samples to create your reference genome and the 7 other ones to do like a mini study like that. This is how these things would look like. So this is chromosome A, chromosome B, your reference, all the high C data plotted against it. If you're getting something like this, this would be a translocation, because how you read this is there are reads that map back in chromosome A, and there are reads that map back in chromosome B, so to a reference. So this is the reference, this is the high C data, and that means a part of this chromosome is now here and vice versa, and that's why this is a translocation. The nice thing is if you do something like that, you can just visualize it and you just see it. For example, I'm not very good in bioinformatics, but I can see this as a structural variation. Oops. Yeah, and you can do this across the genome. I mean, we did this, with human data because it's just easier, because there is a human reference genome. So this is basically all the chromosomes, and you can see all these structural variations here. I think I have it somewhere what these actually are. So this is a translocation, this is a deletion. You can see amplifications and everything. So it is really works very, very well with for comparative genomics. The second thing you can do is you can actually look at transcriptional regulation. As you know, and I mentioned this before, typically you have a linear view of the genome. You have your promoter, something binds to it, and you're getting your gene. But in reality, there might be, for example, things that get recruited to this promoter, for example, this enhancer region, and you can use your Omni-C data to see that as well. So yeah, this is basically an example how this looks like. So you basically see this part here where two things are very close together. Sorry, yeah, where these two things are very close together. The nice thing is if you're doing a genome assembly, and I have mentioned this before, our rule of thumb is you need 100 million reads per gigabase of genome, which translates to about 30 times coverage. So the data set you're using to create your draft assembly will give you exactly this kind of data set. So how the chromosomes folded, the 10 kb bin resolution. If you want to know more about this, what bin resolution so on is, I am actually, we are organizing another webinar next month. I think it is the 14th. I'll post it in the Slack channel, how to use this proximity ligated libraries for these kind of things. But as I said, the nice thing is, even if you just do genome assembly, it is just information that's intrinsically in the Omni-C data set, so you can actually use it for that. Yeah. Yeah, and basically this is how you read these things. So again, reference, all the high C reads plotted against that, and then you can see what areas are close to each other. So A and B, you can see this structure here that shows you that A and B, these two compartments are very close to each other, or even smaller areas like here. And if you want to do this with loop calling, you can do this as well. So again, it's the same thing. We're plotting it against it, and sometimes you see these dots here. This dot basically means that this part and this part of the DNA are now co-located. So if you're mapping this back to this part here and this part here are together. And because we have this more crisp data with Omni-C, we get just way more reads to support a certain loop when we call it. So why is that important? Like generally, why is structural variation important, and or SNPs calling so important in these kind of things, and how does it impact the gene regulation and TAT forming? As I said, we have a talk next month. It goes into way more detail, but just very roughly. So for example, this is a catalyst from a translocation in a cell line, meaning there is a translocation between chromosome 7 and chromosome 14. This is the control. So if you're looking at this part here where the these lines are, is the part that is a structural variation. And you can see here, nothing happens between chromosome 7 and chromosome 14 because they are very far apart. In this event where we have a translocation here, you can see we are losing half the reads because it's haploid. So that means on one allele we have the structuration, we don't have on the other. So you can see that the density, the read density actually goes down. And also in this location, we're getting a new TAT here. So because now these parts from chromosome 7, chromosome 14 are next to each other, we are creating a new TAT. So it's called neo-TAT. It's not really clear what to call this point, but quite a few people are calling this neo-TAT. These are important because usually if you have regulation of a gene, regulation doesn't cross a TAT. So that means if there's a gene here and a gene here, there won't be any regulation here. They are only within one of these TAT structures. So for example, if you have a SNP in a promoter region, this can actually change the chromatin structure in a way that certain enhancer or silencer sequences cannot get back to this promoter region, therefore changing the expression of a certain gene, even though there might not be a mutation in the gene itself. Oops. Yeah. And I mentioned this before, because we have this uniformity of coverage, this library works very well for SNP calling. So we compared, in this case, a cell line GM12878. So this is a human cell line because in this cell line, the SNPs are very well described. So we have a shotgun library, we have the Omni-C library, and we have restriction-enzyme based Hi-C. And we ran it through the GATK pipeline and basically can see almost all SNPs. So as I said before, if you want to do comparative genomics and you are interested in the SNPs, basically create your reference with Omni-C, create Omni-C libraries, and you get not only the SNP data, you get the structuration data and some other data sets on top of it. It's basically included in your data set. And the nice thing is, because you're getting all the SNP information, you can the Omni-C data set is significantly better in phasing. So we did this with HapCut2, and I only have this one slide here. It's actually a full talk, and it was done by Jay Guria some time ago. I will post it on Slack because it goes very deep into the bioinformatics. He's actually comparing the phasing of a human genome plus the Atlantic bluefin, which was done with Stanford University and PacBio. But basically, the takeaway message is that creating a reference with HiRISE and comparing the phases worked really, really well. So you can see here the N50 with 28 megabases on both phases. And as I said, the data set I have there has some K-mer analysis, and it really shows nicely even the differences in structural variations between the two alleles. Yeah. This is now my last slide. This is something that we haven't done before. I know there are a few people working on this. So middle of last year, a Chinese group launched a new pipeline called All-Hi-C, which claims to phase polyploids as well. HapCut2, what I just showed you before, is mainly is only phasing diploids, but All-Hi-C claims to phase polyploids as well. I know there are some people working on this right now because the Omni-C dataset is compatible with All-Hi-C, but I haven't seen any results. So if anyone wants to do this, ping me and we can see if there is something of interest there. Good. So just at the very end, as a last slide, so I'm actually much faster than I thought. I'm 35 minutes in. This is just, again, the promotion we are currently running. The deadline was actually 2 days ago, but we extended it to the 15th of October because of this conference here. So we can advertise this to you as well. If you're interested in this, you can either apply through our website or send me an email or send me something on Slack, and I can send this to you. If you're interested in doing something like this in yourself using the Omni-C kit, I can help you with that as well. We're actually organizing a bioinformatics workshop as well that will teach you how to deal with these proximity ligated data sets, because I know that some people are kind of scared of them, because they do differ quite a bit from a normal shotgun library. Okay, with that, I think my talk is finished. So you have more than enough time to grab a coffee before the next session. If you have any questions, you can just post them on Slack or, as I said, email me. Thank you very much.